Hello class, today we're going to be discussing the Lutheran blood group system. I'm going to be starting off and then Cameron will be finishing the slides. So first, at the end of this lecture, we would like you to be able to describe the clinical significance of Lutheran alloantibodies, the immunogenicity of the Lutheran antigens, as well as the antibody specificity, whether or not the Lutheran system experiences dosage, where on the cell the Lutheran antigens are located, and the modes of inheritance of the different Lutheran phenotypes. We're also going to be discussing the incidence of the Lutheran antigens, the development, or whether or not when they are developed, how early on, any known biological function of the antigens, and we're going to be correlating the Lutheran system with some disease states and discussing how they react with routine enzyme treatment. We'll also be explaining the background and the history of this blood group system, and we're going to be discussing how the frequency affects the clinical significance of the antibodies, and we're going to compare and contrast the characteristics of the different Lutheran antibodies, and again the phenotypes of the Lutheran system. So first, with the immunogenicity, the Lutheran system follows the same criteria as most other blood group systems, meaning that they are RBC immune. For example, the Lutheran B antibody is developed only after pregnancy or after transfusion of blood that is Lutheran B positive into someone who is Lutheran B negative. Those that are able to create the Lutheran antibodies, the antibodies tend to be a mixture of different antibody types. For example, anti-Lutheran A is usually IgM and is reactive at room temperature, not so much at 37 or in the AHG phase. Anti-Lutheran B, however, is usually IgG, though there have been some IgM and IgA antibodies identified, and IgG is usually reactive at the AHG phase. And some Lutheran antibodies have been found to bind complement, though very few examples of in vitro homolysis are there. When it comes to the genetics and the inheritance of the Lutheran antigens, the gene for this system is called BCAM, and it's on chromosome 19. As you may recall, there are other blood group systems that have their genes on chromosome 19 as well. A couple of those that are mentioned here are secretor and Lewis. I mention those because there's been a linkage found between the Lutheran genes and the secretor and Lewis genes. I'm not entirely sure what that linkage is. I'll have to read more into that, but I do know that there are studies that show a linkage between these blood group systems. When the Lutheran the Lutheran proteins are on the cell, they are a type 1 transmembrane protein, and they exist in two forms. One is a Lutheran glycoprotein, and the other is a shorter basal cell adhesion molecule, shortened to BCAM. And here's a depiction of those glycoproteins. The one on the left here, the Lutheran glycoprotein, it has a longer tail inside of the cell membrane. And then the BCAM has a shorter tail inside. Other than that, they're very similar, if not identical. These first three spots are constant regions, and these last two are variable, and that's the same on both ones. It's not so much important that we know which ones are the constant, which one are the variable for this lecture. Though I would like to point out that the Lutheran A and Lutheran B genes, or antigens, will bind onto that last portion, that last variable group on the Lutheran antigen. As well to mention, these 5 are domains that are connected by a disulfide bond, and that'll be important in just a sec. So with enzyme reactivity, Lutheran antigens are not consistently enhanced or destroyed. It all depends on what enzyme we use. When treating cells with physin, papain, or glycine acid EDTA, the Lutheran antigens are resistant, so they will most likely be enhanced because they won't be destroyed. If you treat the cells with trypsin or alpha chymotrypsin, then they will be destroyed. Additionally, and this is interesting, when we have Lutheran antibodies and we are testing them against cells that have been treated with DTT and AET, the antibodies won't react, even if the cells were shown to be Lutheran positive. And the reason that this is because DTT cleaves those disulfide bonds. And as mentioned earlier, the glycoproteins that house the Lutheran antigens are attached to that transmembrane portion by disulfide bonds. So when we treat it with DTT, it is breaking those bonds and thereby causing the antigen to no longer be there. Lutheran antigens have been identified on fetal cells as early as 10 weeks of gestation, though it's shown that to be poorly developed at birth, even when they are there, and HDFN is rare and mild if it does occur. Another reason that is speculated to not cause HDFN and to be mild if it does is because the Lutheran antigens are present on many different tissue types as well. So with that being the case, the placenta is one of those tissue types where the Lutheran antigens are. So any Lutheran antibodies coming through from the mother to the fetus would be taken up by those antigens. So it doesn't stand a high chance of getting through that antigen shield or netting, so to say. Something interesting is Lutheran proteins have a multifunctionality property to them, which means that they don't just act as, oh, look, I'm Lutheran. They can also bind laminin, which is a protein in the serum and on other tissues or other cell types that helps to bind different things. And an instance why this is interesting is Lutheran proteins have been shown to bind the laminin, and when in sickle cell patients, it's more of a problem because the laminin and the sickle cells, they tend to clot and form obstructions in the vesicles, which can lead to problems. This will be discussed further in detail when Cameron gives his portion, but anti-Lutheran A is not usually considered clinically significant unless the antibody panel reactions indicate that it is the causative antibody. But on the reverse, anti-Lutheran B is always considered clinically significant. And again, the reason why should become clear when Cameron is giving his portion.